Okay, good afternoon, everyone. Thank you for joining us today. My name is Billy Wilson. I work for the Department of Fish and Wildlife in the Habitat Conservation Planning Branch. Today's lecture is part of the Conservation Lecture Series. Or register for upcoming lectures and watch videos of past lectures, or subscribe to our mailing list on the website you see on this first slide. Here's a schedule of our upcoming lectures. Next month, you'll see that we have brownwater black swans. In June, we have conservation working dogs. August, we have badgers. In October, we have scheduled in October is for the San Joaquin kit. For today's lecture, I ask audience members, please hold their questions till the end, and then we'll come with a microphone so those listening over the internet can hear your questions. And for those of you listening through the internet, please type your questions into the notes box, and the presenter will answer your questions at the end. Today we are pleased to have David Wyatt presenting a lecture on white nose syndrome in bats. David is a professor in the field ecology program at Sacramento City College. He has taught at Sacramento City College for 17 years and adjunct professor at American River College from 1993 to 1998. Wildlife biologist by specialization, and his ongoing research work has been concentrated on ringtails since 1987, bats since 1996. Prior to his calling in higher education, David was a biologist for the California Department of Fish and Game, now Wildlife, and for the Department of Transportation. He has been working since 2002 as a wildlife consultant specializing in bats and ringtails, and leads are the sometimes happy humans to 5 beagles. After all, one of his favorite naturalists is Charles Darwin. Please welcome David Wyatt.
So thank you very much for coming out to hear about white nose syndrome. This is actually one of those weird things where you have, as a biologist, I have this tremendous amount of excitement about something that also gives me abhorrence and fear and just absolute sadness. I'm not talking about the lecture, I'm talking about white nose syndrome, that it is a disease that, as a biologist, excitement in that we have very rarely occurrences where something so devastating to wildlife ever pops up during our lifetimes in particular. So as we have the ability to actually study this developing and infectious emerging disease within an entire wildlife group that has the ramifications in terms of conservation biology of affecting vast areas of ecosystems, not to mention human health impacts and human agriculture impacts, we very rarely see something like this. So that's the excitement part, while tinged with that is that abhorrence with what it's doing to our wildlife populations, especially of the hibernating bat populations around the eastern half of the United States, and the fear that's associated with what's going to happen here in the West in terms of detectability, as well as our reliance in California on bats for so much agricultural pest control services. How are we going to address that? That's actually a very important type of question. I think one of the most startling things that you find out about when you're in the bat world and you subscribe to journals like Bat Research News or other journals like that, hearing about the PTSD that a lot of the biologists back east are suffering because they go into their Hibernacula where they counted 186,000 bats 4 years prior, and then all of a sudden finding 1,500 or 2,000 bats, and the floor of the cave is littered with bones of the bats that have died. And Tom Coons was telling the story, and you could just hear it in his voice, the amount of pain that was going on at a conference I went to. And his grad students just openly crying as they're pulling out wing bones with bands on them, and as he's reading off the numbers, he's getting the numbers read off, finding out that bat had been banded 15 years prior, or 20 years prior, or 5 years prior, and they're all dead. All the subjects are dead. As biologists, can you imagine if all of a sudden, almost overnight, all of your subjects were dead? And I would have to say that as biologists, we also love our work, and as a result, we care for our subjects, and that would be devastating. And you can see why they go through PTSD. So I think it's very important out here in the West, even though we've been insulated from white nose syndrome for a period of time, that we actually do learn about it and learn about what techniques we can use. And there are some things that are very promising on the horizon. I got to read an article from April 8th in a Prolos journal that gives you some hope that this may be something that we could deal with, and I'll go over that in a second too. So I was struggling with the title. Back when I originally put this together, I had a little bit different title than what's posted on the agenda, but I thought, a review of the progression of the disease and the detection challenges facing the Western states sort of sums it up in a nutshell, what this talk is going to talk about. And as was mentioned, I'm with Sacramento City College, and I teach in the field ecology program. And I see a lot of my former and current students are out there too. So thank you very much for having me here. So let's look at emerging infectious diseases in wildlife. There's a number of these. We have a highly infectious diseases that affect entire groups of organisms like Chytridiomycosis, which affects large numbers of amphibians throughout the world. And it's found on every single continent with the exception of Antarctica now. Myxomatosis affecting rabbits in Great Britain, and they've actually introduced it into places like New Zealand and Australia to try to control the rabbits as they became invasive. Devil facial tumor disease affecting Tasmanian devils, where they become unable to see or eat because of the tumors that are growing on their faces. West Nile virus, which is affecting birds in North America and had this rapid transmission from New York to California, and in less than 5 years it had transmitted across the United States. And then white nose syndrome affecting bats in North America, which was first detected in 2006. Now, out of all these diseases listed right here, 2 of them are fungal diseases, Chytridiomycosis, again a broadly based disease of amphibians, and then white nose syndrome, a broadly based disease of bats, in particular hibernating bats. So let's look at BD. BD is the causative agent for this fungal disease, and we call it Chytridiomycosis, affecting amphibians worldwide. And I just put this picture of a cellular structure because I want you to just note that there's a lot of open spaces in there, and we're going to see a picture of a bat wing which shows very similar characteristics. And what we're seeing is a whole bunch of dead yellow-legged frogs or red-legged frogs, resulting in listing packages that have to be developed as a result. And then Pseudogymnoascus destructans, used to be called Geomyces destructans, is the causative agent for white nose syndrome. And look at those spaces in the wing membranes. So basically it's dissolving away portions of the wing, causing extensive amounts of tissue damage to the bats. Chytridiomycosis and white nose syndrome are both continuing to cause rapid declines in amphibian for Chytridiomycosis and bats for white nose syndrome throughout the world. We find every continent except Antarctica affected by chytrid fungus, but only North America and Europe affected by white nose syndrome. And the European connection is actually a very interesting one. So we'll talk about that in a moment. So looking at these 2 side by side, if you look at chytrid, it was first discovered in Queensland in 1993, while white nose was discovered in North America in 2006. Subsequently, we recognize that it's also in Europe, and that has probably been in Europe for maybe as long as 10,000 years, based upon the amount of mutation that's gone on with the variants that we have of genetics associated with Pseudogymnoascus in Europe. Chytridiomycosis is a waterborne pathogen, while it's soil-borne for white nose syndrome. Genus was first documented of Chytridiomycosis in 1938 with African clawed frogs. And African clawed frogs have 0 mortality due to it. 0. They don't die from it. So perhaps that's where it developed, and all of the susceptible, in terms of evolutionary process, all the susceptible frogs ended up dead, and what you have are the leftover ones, which are completely resistant to it. But they can actually transmit it. And we used African clawed frogs throughout the 1940s as a pregnancy test. So we distributed it among almost every single continent. And they became released out into the wild, and they established populations, and they also transmitted diseases. So some frogs are unaffected, like the African clawed frog. Others have 100% mortality. Where you get it, you die, basically. It's unknown if the current virulence is due to a new emergent pathogen or whether it's just increased virulence that's happened with chytrid fungus. Now, white nose is also a soil-borne pathogen, but also a cold-loving fungus. And hibernating bats are the ones that are susceptible to it, where you can have upwards of 99% mortality in some species. The smaller you are as a species, as individuals, the more likely you're going to be dead from it. Both diseases like cool temperatures. That's why we tend to see Chytridiomycosis up in the Sierra Nevada, although they can survive in lower elevations where it is present, even in the tropics. But in the case of Pseudogymnoascus, it doesn't like the warmer temperatures, so it doesn't really grow. Affected individuals become anorexic after these diseases penetrate the skin membranes and start to basically cause the organisms to leak, and they lose fluid, so they dehydrate, and they spend all of their immune system trying to battle this, that they end up going anorexic and dying. There are no treatments of this in wild populations. We have no ability to treat this in wild populations. And now we're starting to look at bacteria that coexist with them. As you know, we have all this bacterial fauna on and in us that if we did not have that present, we would die. We have to have this community of organisms. And the same way with bats. So we're starting to look at that community of organisms and seeing if that can actually battle Pseudogymnoascus. Fungal diseases are notoriously difficult to treat. I mean, really bad to treat. If you've got athletes, well, you know this whole deal, that it is always coming back. Not only do you have spores present everywhere, but it is just a persistent sucker, and you just cannot get rid of these things. In humans, we haven't been successful in developing effective vaccines against any kind of fungal illness that we suffer from. And we've tried. So they're very difficult to do. You have topical treatments that you can do. You can rub creams onto you and they'll control it. But the problem is the toxicity levels are so high that if you put it on bats, for example, the mortality rate of the treated group is higher than the control group because the toxicity levels are so high. So you're just doing the bat even worse damage as a result. White nose syndrome was first noticed in February 2006 in a cave called Howe's Cave. It's caused by the Pseudogymnoascus destructans, and this is basically what the first biologists saw. Now, the storyline that I've heard, and whether it's true or not, it does sound good, was some cavers had gone into this cave and noticed these bats with this white stuff coming out of their noses. And cavers can probably be some of the best friends we have in trying to detect this early. And they took some pictures of it and didn't think much about it. And a little bit later on, they encountered a gentleman by the name of Alan Hicks in New York State who said, they said, hey, have you ever seen this in bats? And we saw these pictures, showed them pictures, and he thought, what is this? And went into the caves trying to figure it out, but the bats had already left, or it was post-hibernation. So it was really in 2007 that we went into these caves and found this, where we have a lot of this fungal growth coming out of the membranes, out of the noses of these bats, and they were emaciated. And that's when the, we may have a problem here, came about. What was unexpected was how fast it spread. It was first detected on February 17, 2006. It's now found in 25 U.S. states and 5 Canadian provinces as of 2015. And there are 3 additional states in which Pseudogymnoascus has been detected, but white nose syndrome hasn't been confirmed. Cold-loving fungus, found in Europe also. Europe has about 16 to 17 genetic variations of this based on the Portland Wildlife Society meetings. I went to a white nose workshop, and they were talking about 16 or 17 variants of it, while in the United States, there's a 3 base pair difference. Doesn't matter whether you're halfway across the United States or whether you're in New York State. 3 base pairs. So it came from a single infection point from a single variant of Pseudogymnoascus that's spread through the United States now. So it's been present in Europe for a long time to have the ability, or to have that divergence into 16 or 17 different strains. We also noticed that European bats appear to exhibit lower mortality rates. Is it because it's already burned through the population? Probably. I mean, the vast majority of them died. And we do have bats that are surviving white nose syndrome here. So maybe that's the case. There's some evidence indicating that maybe the bats that are surviving are the ones that preferentially seek out, instead of 40-degree Fahrenheit locations for hibernation, the little microclimates in the caves that are at 50 degrees Fahrenheit. And Pseudogymnoascus simply just doesn't grow well at that. It still grows, but it doesn't grow well. And they're able to keep up with it so that instead of waking up every 2 to 3 days, they're waking up every 2 weeks or 10 days. They're able to have enough energy left at the end of the year to go out and forage. There's also the ramifications of climate change. I mean, we have spring arriving 3 weeks earlier and fall or winter arriving 3 weeks later, so are they simply reducing their hibernation period? And why do they hibernate? To save energy. So maybe that might be a good thing about climate change. Yeah, we're going to lose the pika, but we're not going to lose bats. I mean, maybe that's the trade-off. I don't know. Bat mortality can be as high as 99% annually. In the Northeast, the regional mean mortality rate is 75%, 3 out of 4. Now remember, this is a case-selected species that only gives birth to 1 young per year. This is a very, very tough rate of mortality. I mean, we're looking at, essentially, if you look at a timeline, it's a stochastic extinction event. It's happening almost overnight in terms of geologic time. In the Northeast, large hibernacula sites became infected within 2 years of the arrival of white nose syndrome in the region. Why? Bats move around, and they carry the fungus with them, and they move around and inoculate new cave systems with fungus. I was talking this weekend with a bat biologist, Linda Anger, with the Forest Service, and we were talking about Alan Hicks and how we felt bad for him because he had this, it's almost like a survivor's remorse type of deal, because he's the one that made the discoveries, and he felt so bad the first few years because as he's investigating caves, his question was, did I just inoculate a bunch of caves? And so no, the bats are inoculating the caves themselves. So I think he's come to that realization too, and that's part of that PTSD type of aspect associated with what biologists are going through. So in hibernation, what are the benefits? I mean, you're basically reducing the energy needs for a species. I mean, the food needs. The reason they're hibernating is because they don't have enough food available in the wintertime. When you reach temperatures below 40 degrees, the noctuid moths are no longer flying. You don't have midges flying, so they have to find a way to survive. And either they migrate out of the area, like Mexican free-tail bats might do, or they go elevationally up and go into hibernation, or they go into hibernation where they're located. So they're able to survive these cold periods with minimal expenditures of energy. The challenges are they have to initially store enough energy to survive, and frankly, insects are actually a really good food resource, and if they store up enough fat reserves, they're able to survive through the course of the winter. But they have to be very, very conservative in terms of how they expend those energy sources. So you're talking about bats like the California myotis out here, which is 4 grams in weight. I mean, they can't pack on 2 grams of fat. So they have to, a lot of these bats are hibernating on essentially the energy equivalent of 2 pats of butter for 3.5 months, maybe 4 months. So they have to conserve energy very, very well. Do this by reducing certain metabolic functions, and things like the immune system is a good example. So Pseudogymnoascus, cold-adapted fungus, grows well in 40 degrees Fahrenheit. I mean, the reason we put food into refrigeration is not to prevent spoilage, but to slow it down so that we can eat the stuff before it becomes fully spoiled. Because guarantee if you've left a tub of cottage cheese accidentally in the back of your refrigerator, and you don't clean your refrigerator very often, you open it up after 8 months after the expiration date, it's going to be full of fungus. Okay, so it's just slowing it down so we catch up with it. Problem is, Pseudogymnoascus loves 40 degrees or temperature, which happens to be the hibernation temperature for bats. They also like high humidity conditions, and what do you have? You have bats exhaling, and you have higher levels of humidity as a result. So everything's perfect for Pseudogymnoascus. Bats tend to have high humidity conditions within their hibernacula to conserve energy. They turn off a lot of metabolic function. On average, you're looking at something like every 3 weeks a bat would rouse. It would sit there and ramp up their energy, becoming euthermic again, and then it's processing metabolic waste. It's getting rid of any latent infection they have, usually small amounts because most things don't like the cold and grow slowly, and then they go back down in torpor. But when you have white nose syndrome present, they're waking up as frequently as every 2 or 3 days. So instead of waking up every 3 weeks, they're waking up every 2 to 3 days, and a month, month and a half into their hibernation, they wake up with no energy stores left. And they try to fly around, and you'll see concentrations of these around the entrances. They try to go out. They go, wow, it's really cold out here. Come back in. And then finally they just run out of energy, fall to the ground, and they're dead. Okay, that's how we're finding these guys. So they literally starve to death. That's how they die. So when you look at patterns of disease progression in Hibernacula, initially pre-white nose detection, you have some levels of increased populations because maybe other bats are moving from areas that are diseased, going into these new areas, thereby inoculating them. And around the time we first detect white nose syndrome, you have a lot more of activity in the daytime of these bats because they're rousing more frequently. As they rouse more frequently, they're going to go around and go, man, I'm a little hungry, and they go out and try to find food, and there's just no food available. You start seeing a few bats with some fungus on them, actually growing on their noses and on their skin where it's exposed, and you don't see too much in the way of dead bats, occasional dead bats, and that's normal for a Hibernacula anyway, so the population remains stable. Then it goes crazy. Okay, subsequent winters after that, you're having lots of bats showing up with this fungus because there's just simply a lot more fungal spores present. And then you have increased mortality of bats, significantly increased mortality. And then the population counts start to drop off the map. And off-site mortality is something we don't know as much about, unfortunately. So that might be another possibility too. You know, when I went back to what Tom Coons was talking about, I remember him saying, you know, this was at a Denver North American Symposium on Bat Research meeting, and we had nothing scheduled for that evening, and he said, does anyone want to sit down, we'll talk about white nose syndrome? And it's like all of us bat biologists were like, yeah, we want to hear that, you know, talk about that. About 300 of us showed up to this meeting, so it was big, you know. And we showed up, and we remember him talking about going in, I want to say it was like 2005 or 6, counting one cave that he's been working in, 186,000 bats in this Hibernacula. Going in, counting up the clusters of bats, having about 186,000 bats in it. And then going back in 2010, 4 years later, and finding 2,500 bats. And just the ground littered with what he described as it looked like little toothpicks strewn all over the ground because those were the wing bones of all the bats that had died the subsequent winter. That's shocking. I mean, that's just an amazing rapid death rate that actually affects the entire population. So one of the things that's diagnostic about Pseudogymnoascus is these comma-shaped conidia. These are what produce the spores. And notice they've got sort of that little comma shape associated with them, almost like a little garlic clove. And you also see this depigmentation on the wings, but not always. And you see the visible fungus growth on not only the wing membranes, but also on the muzzles of the bats. And subsequent winters, you find huge amounts of population dead. Also, the tissue damage is fairly significant. You look at those arrows, and you can see those little holes in the population or in those bats that are on the lower left. And there are gaps actually in the tissue where the tissue has been destroyed. As you look more carefully using electron microscopy on the surface of the wings, you can see all the fungal hyphae penetrating where the epidermal tissue is located, penetrating into the tissues of the bat, causing rapid cellular death. So how does it destroy tissue? That's a good question. And this was published October 21st, 2013. What's so great about white nose researchers, we decided to publish pretty much everything open access. We don't want to have someone have to pay $25 to get an article. You know, how often do we see that? Or have to be subscribing to Bio1 or something like that. It's all open access under Prolos. And there was a paper that was put out by Radaba and Miller that's a nutritional capability of and substrate suitability of Pseudogymnoascus destructans, the causal agent of bat white nose syndrome. And what it found was it can break down pretty much all complex carbohydrates, or all carbon sources, all complex carbon sources. So anything that's carbon-based, it's going to be able to break down. And though it can grow in a wide range of pH, except it doesn't like growing in acidic conditions. And what it does, it produces a very alkaline material, very similar to getting a chemical burn that's like a base chemical burn. And that's what's actually destroying the tissue, liquefying it, allowing the fungus to feed. But the study also found that PED is very temperature dependent, and at 68 degrees Fahrenheit stops growing. So maybe we have severe climate change and we'll deal with this, but otherwise it's going to be around in Hibernacula, and it has difficulty absorbing water is another characteristic of it. Another paper came out in June 20th, 2012 by Deanne Reeder and a whole bunch of others about frequent arousal from hibernation linked to severity of infection and mortality in bats with white nose syndrome. So you're going to see a little bit later in this program a picture of a data logger that's been glued to the back of a bat. Yes, we can take the data loggers, trim off whatever excess material, make them really light, and then stick them on with surgical glue or something like that right onto the backs of bats. And they did this with a total of 83 bats, or 504 bats that they were looking at, free-ranging bats, and then they would go out and recover the data loggers either from the living bats or the dead bats or data loggers that fell off and document what's happening to these bats. And what they found was during hibernation, the bats that have white nose syndrome frequently roused and became euthermic during the period of their hibernation. And the ones that they found dead, essentially those guys that ran out of energy, they were waking up every 2 to 3 days and becoming euthermic when normally they wouldn't become euthermic for 3 weeks. And that way they were able to save energy. These guys weren't able to, and they died. So this was a really important paper to actually come out. So looking at the epidemiology of white nose syndrome, the earliest case was September 30th, and that was only histological. There was no evidence of white nose syndrome on the muzzle or on the wing membranes. It was all confined within the tissues themselves. The latest known case was June 1st, and again, that was histological. So it's only in the deep winter, in the deep cold, that you see all those fungal hyphae actually going external on the bat. The incubation period appears to be somewhere around 2 to 3 months in length, but within 2 months you can have death very easily. So if you have hibernation starting in December, oftentimes you see lots of dead bats starting around February, maybe into early March. Throughout the hibernacula, you'll see bats with visible fungal growth on them, and no one's immune to this. Their juveniles can get it, older individuals can get it, and individuals that survive white nose syndrome get it the following year because there's no immune response. So there is no way to make a vaccine on this because their immune system's turned off. So they simply cannot deal with it in a manner that we would normally expect. There's no apparent sex or gender selection. Scar tissue is oftentimes depigmented, but that is not diagnostic because other things will cause depigmenting of their tissues. And supportive care does seem to work. So if you do have bats that are undergoing problems with white nose syndrome, you can take them out of the colony. Now, granted, you're running a risk of spreading the fungal spores. And you can give them palliative or supportive care, and they will actually survive it. But then they're just as susceptible the next year. One thing that's been really weird that's come about is Meyer found immune reconstitution inflammatory syndrome. This is where if a bat survives white nose syndrome, comes out in the springtime, and their immune system has a reflexive or reflective immune response that starts to act as an autoimmune disorder, and they send out so many antibodies that it starts to overwhelm the organ systems of the bat, causing organ system failure. And the only other group, it's never been seen in wildlife except for these bats, and the only other group of organisms that has this happen are late-stage AIDS patients with humans that do this. Wow. Is this convergent evolution or what? This is really interesting, aspect. And Meyer has this great quote that I had to quote. It's cellular suicide. The immune system comes out in a huge wave going out to those areas of infection and kills everything. It doesn't matter whether it's cells of the bat or not. It just has this haywire response. And it's only been found in AIDS patients or in bats affected with syndrome. A scientific paper was published in April 8, 2012, which also strongly suggestive of fungus being brought over from Europe, and it's pretty much a done deal. We know that. And the question is, how did it get over here? And a lot of these cave systems where we see white nose syndrome in Europe also have a lot of tourist activity. And the first cave that this was found in, Howe Cave, gets 50,000 visitors a year. So equipment and shoes and clothing is probably how it ended up here in the United States. And as a result, now we see it spread by bats. And this is what we're seeing, bottom of caves. We're not necessarily going to see that out here in the West, though. So the West is a very different situation. As of 2015, white nose syndrome is affecting 7 species of bats in 25 U.S. states and 5 Canadian provinces, but 5 additional species have been found with the fungus. So they haven't been, yes, they have white nose syndrome, but it's pretty darn sure, yes, they have white nose syndrome. None. So the epicenter, David Blenner produced this diagram. You can see Howe's Cave, Schoharie Cave, and Hales Cave were the first 3 caves, and they're all tourist caves where it occurs. And then from that point on, it just spread. And here's a good graphic of how it spread. 2 years after initial detection, it was already found in 5 states. It had moved 5 states in 2 years. A year later, it was in 9 states, and 2 years later, might as well stop counting. Look how it went right down the Appalachian Mountains, went up into Canada. All of those bright red spots are new areas of occurrence. The Oklahoma one was, they thought that they found, at that time called Geomyces destructans, they thought they found Geomyces destructans. Turned out it was a different Geomyces. It just had the false positive on it. So you can see the black in New York State, in eastern New York State, is where it started off. A year later, all the purple are the new counties where white nose syndrome, not Pseudogymnoascus destructans, but white nose syndrome was suspected or confirmed from. Then they switched to blue because they're running out of colors. And blue became the new every year. You can see how it's moving. I mean, going backwards, look at the number of counties getting affected. And then 2013, new blue areas. 2014, all those new blue areas. Going down into the south now. It's down in Mississippi. You can see there's quite a bit of it. All throughout Newfoundland, Nova Scotia. Then 2015, March 12th, all the blue areas. But I want you to pay attention to upstate Michigan because just 3 weeks later, you can see they added new areas. See, I'll flip them back and forth. So we added new areas where white nose syndrome was confirmed 3 weeks later. So it's visibly present all throughout the eastern half of the United States, and it's had devastating impacts. Now, the affected bat species, the 7 that are affected are big brown bat, gray bat, and endangered species. Eastern small-footed bat, little brown bat, northern bat, which is a threatened species, just got listed by the U.S. Fish and Wildlife Service. Indiana bat, which is an endangered species, and the tricolored bat, which is probably going to get listed. This one's taking some big hits. The other ones which have been found with PD are the Virginia big-eared bat, which you're going to see that guy listed. Rafinesque's big-eared bat. If these guys come down with white nose, their numbers are already, down in the dumps. Southeastern bat, silver-haired bat, which was really something we didn't expect. Okay, and then the eastern red bat. This is a foliage-roosting bat. How do they come in contact with this? It's a good question. Are they socializing with other bats? I don't know. The western red bat seems to be pretty much solitary. So what's going on there? We don't know. Northern California bat species, we have Anthrazoa pallidus, state species of concern. Cornurinus townsendii, candidate for threatened. Big brown bat, Tuscus fuscus. Euderma maculatum, spotted bat, species of concern. Lasionypterus noctivaga, and silver-haired bat. But of the small bats, we got California bat, western small-footed bat, western long-eared bat, little brown bat, fringed bat, species of concern. Cave myotis, long-legged bat, species of concern. Yuma bat, canyon bat. 1, 2, 3, 4, 5, 6, 7, 8, 9 species that are small. And to give you an idea of size-wise, this is about, if you look at them side by side, those Parastrellus hesperus, the canyon bat on the top, we got a California myotis on the bottom, and right in between we have little brown bat, Myotis lucifugus, and we have a pallid bat on top right and a big brown bat on the bottom left. So we're seeing things like big brown bats having about a 60% mortality rate back east, while these little guys having around 99% mortality. They just simply don't have enough energy to survive waking up every 2 to 3 days, while these other bigger bats might. So that's another wrinkle because we have a lot more species than they have back east of small-sized bats. They looked at population trends of little brown bats in northeastern United States, and you could just see how it drops off the map at 2010. Some sites were affected in about 2000 or around 1995, 96. That was major flooding events they had in the caves, which ended up higher mortality as a result. But, they recovered from that until white nose syndrome. And that gray area to the right of each of those graphics is the period of time in which white nose syndrome was first detected to the death rates. And if you remember, it's about 2 years after first detection that you see the collapse in the numbers of the bats. These are 42 hibernacula in New York, Pennsylvania, Vermont, Virginia, and West Virginia. And they looked at mortality rates. The sum of pre-white nose syndrome, little brown bats had about 350,000 plus. After white nose has started to kill off bats, they're down 30,000, 91% decrease. Northern long-eared bat, the one that just got listed, 98% death rate. Tricolor bat, 75%. Indiana bat, which is an endangered species, 72% death rate. Eastern small-footed bat. Why is that one not affected? We're not sure. Maybe it doesn't like to gather into clusters or something, so it's not transmitting very easily. 12%. Big brown bats around 41%, but overall the average is 88% of all the bats that we had counts on died during this period. So what about the economic impacts of white nose syndrome? And between 2006 to 2012, it's killed somewhere close to about 6.5 million bats. In April 2011, a study was published about the value of bats to agriculture and the economic impacts. And we're looking at a range of somewhere around $3.7 billion of free pest control for us to a high of $53 billion per year, and that noticeable losses to the economies of agricultural regions of the United States are to be expected within the next 4 to 5 years as a result of threats to bat populations. In California in 2010, we had $40 billion in revenue generated in agriculture. A single little brown bat, which has a body no bigger than an adult's thumb, can eat 4 to 8 grams, the weight of about a grape or two of insects each night. Although that may not sound like much, the loss of 1 million bats in the Northeast has resulted between 660 to 1,320 metric tons of insects no longer being eaten each year by bats in the region, and that went up to around 6.5 times that. Bats eat tremendous quantities of flying pest insects, so the loss of bats is likely to have long-term effects on agriculture and ecological systems. Just look at an airline flight. I mean, this was a picture I took out of a flight going to Salt Lake City, and I just snapped off a bunch of pictures. I know you're not supposed to use electronics on planes, but oh well. So I got a picture of the actual farmland. You can see the Central Valley is crisscrossed with farmland, and as a result, bats play a very important role in our economic development. Rick Adams is a bat biologist working out of the University of Northern Colorado, and he has some great statements. This is one of those situations where humans are definitely going to see the effects of it. It's like your health. You don't think about it or notice it until it's gone. Bats pull tremendous numbers of insects out of the air every night, and we don't thank them for that often enough. When they disappear, people are really going to start noticing. If it gets into the west, it's really going to accelerate even further and affect so many more species of bats. Ecologically, it would be an Armageddon type of scenario. Now, maybe that's some hyperbole, or maybe that's true. We don't know what it's going to do out here in the west. Why? Well, look at the number of bat species back east versus the number of bat species in the west. The green is showing you 13 to 16 species, and then that's the dark green, and the lighter green is 10 to 12 species. You have very little of that kind of bat diversity in the eastern half of the U.S., and there's a simple reason why. Look at the topography. If you want to look at topography back east, you're running into caves and mines at very low frequency of occurrence. So you have things like Myotis lucifugus formerly occurring in colonies of 200,000, 250,000, 300,000 bats, while the same species out here in the west occur in colonies of 20, maybe 200 if you're lucky. That's a pretty big sized colony for us. Why? Look at the amount of habitat that they have, that they can occupy. They're much more diffusely spread through the environment. California alone has a tremendous amount of topographical variability. I mean, hibernating western bats don't aggregate in very large colonies. They simply don't. Western bats are spread more throughout the environment than day roosting locations than the eastern bats are. We have more caves and mines than the entire east has. California is estimated to have 10,000 open hard rock mines. 10,000. That's more than the entire eastern half of the United States has. Small colonies of white nose syndrome susceptible species make detection of impacts so much more harder. I mean, you can go into a colony with 200,000 bats and go, wow, we're down 2,500? You can see that. But how do you go into a colony of 40 bats and go, has there been an impact? Because you don't know if there was 400 bats there before. And you don't know if something happened there where people were going into the cave that the other bats just left and went someplace else. Because oftentimes they're flying around trying to find food to survive, and they die on the wing, and we don't see them. The top two pictures are what you'll find back east, or used to, and what we find out here in the west are the bottom three, much smaller colonies. So detection is going to be a very, very difficult issue. So we can put bats data loggers on them. These are some examples of some data loggers on bats where they're doing temperature data logging, and bats seem to be able to take that pretty well. Radio transmitters, each of these radio transmitters units weigh about, I was just talking to Fred Anderka from Hollow Hill, and he's got one that's 0.38 of a gram. 4/10 of a gram, and it lasts for almost 2 weeks. It's awesome. The ones I've got are 0.55, and I'm like, Fred, you've improved them since 2 years ago. So those things go on with surgical glue, and they groom them off in 2 to 3 weeks, easily less than that. What about documenting scar tissue on wings and unusual behaviors? You see bats crawling around in the snow, you got to think that's an unusual behavior. You got to figure out what that might be. Please keep in mind, too, that any mammal can carry rabies, and unusual behaviors might be indicative of not white nose syndrome, but something else going on. And rabies being 100% fatal if not treated is a big issue. So anyone that's going to handle bats must be cognizant of that and also have their prophylactic rabies vaccinations. So what else are we doing other than just sort of keeping an eye on these guys? Well, we're taking a lot of samples of bacteria and other things that might be associated with them on their membranes. We're continuing to do surveys where you got to dress up in a full Tyvek suit system, and thank goodness you're going out into Hibernacula, which are nice and cool. Can you imagine having to do these kinds of surveys with a full Tyvek suit in the middle of summer? That'd be pretty miserable. So that's one saving grace, and we just continue to count the bats and still do histological work as their bats are submitted to the U.S. Wildlife Diagnostics Lab. What about use of bacteria as a probiotic? If you haven't read this and you're at all interested in this, you have to get this article. It is so hopeful. It came out April 8th of this year, just last week, 6 days ago. And it describes results of studies of bacteria species affecting PD. And they found 6 bacterial isolates in the genus Pseudomonas that significantly inhibited growth of Pseudogymnoascus destructans. Cool. I mean, you know that in the medical field we use Pseudomonas to treat some of disorders that kill really bad bacteria on us. At the same time, Pseudomonas, other species, can actually give us disease, so you have to be aware of that. Pseudomonas also is a motile bacteria. It can move around. And these isolates were naturally occurring on bats, and they tested them. And what they found, 4 species of bats from eastern North America that were positive for white nose syndrome. These were isolated, and they found that 2 of these species really significantly inhibited the growth of Pseudogymnoascus destructans. So it allows them to survive hibernation potentially. So do we come out here in the West and we seed our caves with Pseudomonas? I don't know. I mean, this is brand new stuff. There are currently few management options to reduce mortality of bats in regions affected by white nose. We don't have really any options. Antifungal drugs are too toxic. It kills them. And other chemical treatments haven't been done in situ, on site. So as a result, we don't know what the impacts are going to be on those. So is it going to be a probiotic solution to this? Well, it's going to factor into it. It's going to be a part of this. But one thing we don't know anything about is what bacteria do we have associated with our bats out here in the West. We don't know that. So that's the next steps that we need to get funded studies that address the bacterial species we have here. Acoustic monitoring is another thing. Now, how can acoustic monitoring detect white nose syndrome? Well, if you do a pre-monitoring process where you come out here and identify areas where certain hibernating species of bats frequent, and you do this long term, so that you get a good baseline of what bat species exist. And then you go out 1 year and you find no myotis. You run it another week later, still no myotis. That's when you start asking questions. That's when maybe it's only a few myotis. You go over and you start to misnet those areas. Every myotis you catch, you use white nose protocols. All of my students get trained in white nose protocols, and we should be training everyone out here in the West for that. And you swab them and you submit that and see if there's PD is present on it, and that might be the only way we're going to have early detection. Otherwise, our early detection is going to be a matter of, hey, where'd all the little brown bats go? They're all dead. Why? 2 years ago, white nose must have shown up here. We can do that using these acoustic monitors, and you put them on tops of cars and you drive at 20 miles per hour. I've established 9 transects, and these are 7 of them, that are 30 miles long. Notice that they're pretty much straight line set up, and they go through habitats. And once you actually have a GPS unit attached to it, it takes a GPS coordinate every second. You match it with a timestamp on your echolocation call that you've identified to the timestamp on the GPS coordinate. And what I do is I just track it in Google Earth. I just dump that coordinate into Google Earth, and now I have a record from that run. I can actually stack all the runs if I wanted to. I can actually filter it for just Myotis californicus or Anthrazoas pallidus or whatever I want, and see what's happening over time on these areas. Of course, I'm not getting paid for it, but so that's one of the things that you have to be aware of too. The other thing too is you can use ultraviolet light. Now PD glows yellowish to yellow-green under UV light. So the top left one shows pre-white nose syndrome infection under UV light, but down below is post-infection. So you can see the remnant fungal hyphae glow. This was a really interesting project that was funded by crowdfunding. And if you haven't been exposed to experiment.com, I encourage you to take a look at it. There's some amazing work being done by people that are fundraising on experiment.com, and I fundraise a project in Belize that way, and they're wonderful people to work with. So I know that's an advertisement, but they are really good. These are some examples of some white nose syndrome detection or PD detection in bats. So I've included this in my kits now. I put Blacklights in all my kits, and every time we catch bats, we run that through, because who knows if there is a fungal species here in the West that mimics this, that does the same thing. We just don't know. We know it works for PD back east, but what do we have out here in the West? So I think it's very important that we maybe run Blacklights on these too. What about building artificial hibernacula? What does that do? Well, you can actually take these artificial hibernacula and clean them. You can treat them with 10% Clorox solution every year, so it doesn't eliminate the potential white nose syndrome transmission, but it certainly reduces it because you have less fungal spores. And they did this with the Nature Conservancy, built this back east, I think it was in Tennessee, and they've successfully had it colonized, and they go in, and after the bats leave, because hibernacula are basically cave systems that slant slightly downward, so it pools cool air. And then summer and maternity roosts angle upwards because they pool warm air. So you have different characteristics associated with cave systems. They made this slanting downwards, so it's going to pool large amounts of cold air, so it became a hibernacula, and Nature Conservancy treats it with Clorox solution when the bats are gone, and they probably harvest, well, I don't know if they harvest the guano, because guano is a great fertilizer, so they can sell it, but I don't think you'd want to do that if you have potential PD present. So they're going to try that out, see how that works. We also have a White Nose Syndrome National Plan. It was started in May 2011. It was developed, and it's been distributed to various states. Federal, state, tribal, and private entities are we're involved with this. And there are 3 major aspects to it: tracking the geographic spread of white nose syndrome, identifying new susceptible species, and then minimizing transmission risks to bats. So they're developing at USGS Fort Collins a national white nose syndrome tracking database. And it will have federal, state, and tribal agency users, so it's a central repository for all this information. Secure database too, so you don't have just general public or anybody getting in there and going, I'm going to go check out this gigantic colony of hibernating bats. You don't have that happening either. There's also the website whitenosesyndrome.org. It is an outstanding website. It gives a lot of the recent literature that you can oftentimes get for free. The National Plan is also posted on this, and they have some really nice posters that you can download and print up, which gives more information about white nose syndrome. So in terms of surveillance epidemiology, we got active surveillance, which is going out and going into Hibernacula, looking at individual bats and taking samples from them, maternity colony surveys, see if they're stable or not, capture studies, acoustic surveys. The advantage is you can probably get an earlier detection because you're physically going out and sampling. It's targeted sampling also, so it's places that you know. Population environmental information can also be gathered as you do your surveillance. Disadvantage, costs money. Okay, costs money and personnel, and that's something that we are lacking in. It's time consuming, it's invasive, it has the potential of introducing PD if you're not, it says GD, sorry, PD if you are not wary, and you can introduce it to new areas. Passive surveillance would be simply responding to citizens responding about bats being active in wintertime, or of cavers saying, hey, I saw something weird in this cave system, wildlife rehabbers getting depigmented wings, animal control agents, non-invasive, inexpensive, cheap, but you also have some of the accuracy and other measures that might be difficult to quantify. You get it bias samples, unknown points of exposure, and also you have transmission too that connect that. The North American Bat Monitoring Program is a program that's recently been implemented by California Department of Fish and Wildlife by Scott Osborne and Scott Newton, his biologist working with them. The North American Bat Monitoring Program establishes a national program to monitor and track bat populations in North America. Essentially what it does is establishes 10-kilometer by 10-kilometer grids of potentially suitable bat habitat, and then a selection criteria goes out in those where they establish upwards of a 22-kilometer long transect for mobile surveys or of surveys of stationary acoustic detectors. And this is a high priority within the detection system. And Susan Loeb is the principal person that's running it through U.S. Forest Service. And there's a number of people involved on a national standard. And a large number of states have already come on board, and California is now coming on board with this. So please investigate that. And if you're California Department of Fish and Wildlife, please support this as a biologist, because, again, the part is it needs to have people to actually do this work. So if you're a supervisor, please be friendly when they call you. White nose syndrome surveillance in Western North America, Hibernacula are typically less than 25 bats. You have topography that makes accessibility to these Hibernacula difficult. Look how much mountainous terrain we have, and there's only so many bat biologists. The West has so many abandoned mines. How do we detect them? Many western bat species hibernate in non-cave structures too, like buildings. What's the potential transmission of PD in buildings? I don't know. Not all normally hibernating species hibernate out here either, or they maybe have different hibernation protocols that they undergo. Not much is known about wintering behavior throughout this. This past winter was a warm winter, and we had populations of bats that were here all year. They never left. The bat boxes that I've run, they were still occupied all this year. And we're talking about Mexican freetails, which are non-hibernators, so they're normally migrating, but they stuck around. Why? There's food resources available. I had pallid bats and also California myotis in the same, and they will hibernate, not always. So they just simply did not hibernate this time of year, or this year. Other questions in the West, I mean, there's lots of questions because they have different behavior, different environmental conditions out here, more varied roost sites than eastern bats. I mean, could these small colonies be less susceptible to white nose? I mean, maybe is that why it takes 2 years for these massive death rates to occur? Because you have to reach a certain prevalence of fungal spores? If small colonies arouse from torpor more often in some areas, will this result in resistance? It's a good question, because they are having an immune response then, maybe more frequently. Will PD persist in non-cave habitats such as cracks and crevices? I don't know. Are there geographic barriers or hostile ecological checks that might inhibit the spread of PD? Is there another variety of Pseudomonas or something else out here that ends up loving to eat or consume or destroy or inhibit Pseudogymnoascus? Will some western bat species show resistance to white nose? How will a drying and warming climate in the west affect bat behaviors involving hibernation, migration, etc.? These are all unknowns. Some of the recently funded research deals with growth and survival of PD under various conditions, also looking at immune response of hibernating bats and post-exposure response. Now, the immune response has been uniformly none, until they rouse. Behavior and physiology of hibernation, population level impacts, both local and range-wide. Genetics, is there a predisposition to some species? Is that why some Myotis species back east don't have the high death rates that other species do? Why is it that tricolor bats have 75% death rates while Lucifugus has 99% death rates? Disinfection or control of PD, including in the environment, the dynamics of transmission, epidemiological modeling. How do we model the transmission of this? Also improving detection of Pseudogymnoascus on bats and in the environment, and then what's the potential for resistance? Does climate change help us in that regard too? So what can we biologists do? I mean, get the National White Nose Syndrome Plan. Go to whitenosesyndrome.org and get it. Educate people about this, professional, other biologists, amateur biologists. They can be our eyes and ears. Cavers, they're great. Wildlife rehabbers. All of these people can be involved in trying to figure out this a little bit better. Take pictures of suspicious fungal growths or scars on bats. I always have my camera handy when I've got bats because I want to take pictures of scarring. Because remember, depigmentation isn't diagnostic, but it could be involved with white nose. When handling bats, use appropriate techniques to prevent contamination. Change your gloves all the time. Do not put more than one bat in one bag. Launder your bags under high heat temperature water. Do everything you can to prevent transmission of this. Disinfect all equipment, Clorox 10% solution, Formula 409 antibacterial cleaner, Lysol quaternary disinfectant cleaner, Lysol disinfecting wipes, or submerge equipment for a minimum of 20 minutes in water greater than 122 degrees Fahrenheit. That's part of the protocols. And then if you have unusual observations, report them to your California Department of Fish and Wildlife office and U.S. Fish and Wildlife Service. And at the same time, I would ask that those people that are getting these reports pass it on to the appropriate people involved with bat work. How the public can help is encourage your state and federal legislators to allocate funding towards the effort to understand and fight white nose syndrome. Federal government has been very reticent in sometimes in terms of Congress of funding white nose syndrome research, and one of the champions that we had died, and it was the former senator from New Jersey, and he passed away, and he was our big advocate for funding of white nose syndrome research. Report unusual late winter bat behavior, such as bats flying during the daytime in snow areas, bats crawling around the ground during winter, etc., and reporting unexplained bat deaths to your state agency. Adhere to state, federal, and local cave advisories and closures to prevent transmission of white nose syndrome, educating our friends and families about the benefits of bats and the white nose syndrome crisis, and following established decontamination protocols. So I want to thank the California Department of Fish and Wildlife for this conservation lecture series and joining also the North American bat effort. I think those are very important things to do to bring out more knowledge about this disease, especially since it hasn't really arrived here yet. The many agencies and organizations that have joined, I mean, they've done this amazing job of coordinated response to understand and address white nose syndrome and to coordinate their activities. Bat Conservation International for being the public face to the general public about white nose syndrome and spearheading educational efforts. To Dr. Merlin Tuttle for now retiring from BCI and taking on the role as the public figure that's going out and teaching people about white nose. And frankly, I got to thank him for getting me into bats and taking the first class with him and catching 565 bats in 3 nights of 19 different species. That'll do it to you, okay? That'll get you hooked. All the biologists working to study this disease, and got to give a toast to our furry flying subjects. There's a couple of pertinent websites here for you as well. Okay, so those are some of the challenges facing the western states. You guys willing to hang around for another 12 minutes? Okay, let's see a video. So let me pop over to that. Go to this one. what do I do with sound? Oh, okay. It was just music there, didgeridoo stuff. Merlin hasn't started talking yet. This is a video put out by USGS as well as this media service that is pretty hopeful, because the first video called The Battle for Bats, you were looking at it going, oh my God, this is terrible. And then since that time, they've come up with some potential treatments and other things that they can do to actually help. And there's also a lot of education effort that's going on that actually benefits bats. And it's a free video. If you go to Bat Conservation International, you can go to their resources link and then go to white nose syndrome. And down below it has a Spanish version of this as well as a standard English version of it. And you could download it as an SD file or an HD file, and you keep it, present it to others if you wanted to. All of that is available. It's about a 12-minute long video, so it's not that long.
Build off mute.
Hey, we can
put the mic down to it.
Oh, okay. That works.
Bats are really incredible animals. They play key roles in numerous ecosystems around the world. In all these ecosystems, bats are critically important in pollination and seed dispersion. And the world would be buried in insects if it weren't for bats to keep them in check. But there's never been a time in history when bats need help more. Because white nose syndrome thus far has been one of the most devastating afflictions to hit American wildlife. It's killed off millions of bats in the Northeast. It's moved south and west, and this is a time of extreme stress for bats, but we now know that even the hardest hit species are surviving in small fragments of the original populations, and those populations we hope will be resistant to WNS.
White nose syndrome hit the state of Vermont in 2008. And at that point, we really did not know at all what we were facing. By walking up to these caves and seeing these bats flying out of these caves and mines here in Vermont in the middle of January, where the temperatures are far below freezing. Those bats were not going to make it. They were dying right before our very own eyes, and we didn't know why. But today we're going to enter the target cave for the research associated with survival from year to year of the bats that hibernate here. And we'll weigh them, determine their species and sex, we'll look for the fungus, and we're going to band them. With a band that is specific to this year. And the idea is that these target research sites will be going to year after year and seeing what we're getting for band returns. And that will tell us that there are some survivors that we need to pay particular attention to. One of the positive things that we're seeing right now is that we're going into these infected caves and mines, and the bats are actually behaving normal. These bats look healthy, and that means that there were some survivors. And these animals have been exposed to white nose syndrome for several years in a row, and that's a good sign. And maybe that's related to their ability to survive white nose syndrome.
So the Fish and Wildlife Service is here with Scott Darling and his team because we are scrambling to try to come up with some management actions that we can take to slow the spread and hopefully recover the bat populations. And we're here to do what we're calling a survivor study. We're looking at the remnant populations of little brown bats that we still find in the northeastern U.S., and we're hoping that what we're seeing here are the seed of a population that will ultimately restore the populations of little brown bats to a viable status. Prior to white nose syndrome's arrival, bats like the little brown bat were extremely common, so common in fact that they didn't warrant counting. The unfortunate reality is that right now we are facing potential extinction of species from this disease. The Fish and Wildlife Service is reviewing the little brown bat, the northern long-eared bat, and the eastern small-footed bat for potential federal listing under the Endangered Species Act. And there are other species that are affected, like the tricolored bat, that are showing signs that they could be in jeopardy of extinction. Today's visit was a good one. We handled about 76 bats. The fact that we were seeing bats, healthy-looking bats, good weights, not a lot of fungal growth, was a good thing, certainly. We have some examples of survival. We've seen it at this site and several others, but really figuring out what that means at a population scale is a critical thing. Nothing is off the table at this point. We're really looking at all angles. We have a national plan in place that has a broad partnership across state, federal, and tribal agencies. We're now rolling out the implementation portion of that plan, and that really lays the groundwork for the entire coordinated response. Because as long as there are bats out here, as long as they're continuing to be affected by this disease, we're going to keep looking for an answer.
The focus of our work at USGS with white nose syndrome is to both understand the physiology of the pathogen that causes this disease and to develop practical diagnostic tools to facilitate diagnosis of the disease and evaluation of management. When we grow cultures, we put a piece of bat wing on a petri plate, and that growth medium elicits the growth of numerous fungi off of that piece of skin. There's something on the middle of that piece of skin that looks like a colony of white nose syndrome fungus growing. And once we have it at this stage in the process, we can then transfer it to additional plates to generate larger amounts of biomass that we can use to study. Fungi can be very challenging pathogens because they reproduce by producing environmentally resistant spores. And so if you have an outbreak of white nose syndrome in a cave, no sign of lesions, no fungus on the nose right there. Now I have it in Illinois. It's very possible that the fungi that are killing the bats are leaving behind literally billions upon billions of these spores. And so the fungus can persist long-term in the environment. And so therefore I think as humans our number one responsibility is that we modify our activities so that we don't inadvertently transmit the fungus around the world. And so that means when we do go into that site that harbors spores of the fungus, it's important that we take basic universal precautions so that we don't transmit it on our coveralls that we might wear and inadvertently make the problem worse.
Mammoth Cave has major bat roosts, and we have something on the order of 400,000 people come and visit Mammoth Cave every year. And now that Mammoth Cave is among the sites that are white nose positive, we were worried about the potential for us to have visitors that can transport white nose syndrome spores to other places. So we've been very proactive in limiting the spread as much as possible as we can by simply having our visitors walking over about 16 feet of carpet and then walking over a mat with a solution to increase the cleaning of the shoes that helps remove the spores, because we don't want our visitors to be the ones who take white nose to Wind Cave or Jewel Cave in South Dakota to wherever they're going home to. And I think the biosecurity maps that we're using are probably one of the more effective means for dealing with walking tours of large numbers of people. I think there's a role for people in protecting bats. White nose syndrome is one piece of it, but bats need a lot of friends. So even if people can't do anything to cure white nose syndrome, the bats that survive are going to need good, healthy habitat, and we need to be doing a better job protecting our cave bat roosts, because bats are an important part of this ecosystem in maintaining it like we see today.
The Forest Service is interested in white nose syndrome because healthy forests need healthy bats, and vice versa. And there are a lot of things that people can do to help bats. And one of the first things you can do to help bats is to provide bats with shelter, like a barn or whatever structure they can use to raise their young in the summer. And also bats like to live in both live and dead trees. So you just want to keep as many trees up on your property as possible. You can also create a home for them by building a bat house. For bat houses, placement is very important. And so bats like it to be nice and warm. So if it's really about the placement and how it's going to control the microclimate that's within that bat house.
One of the great things to come out of white nose syndrome is that we've now suddenly recognized that bat populations need monitoring just as we've done for many decades for birds. And there are opportunities where the public can get involved in helping go out with bat detectors to monitor the status trends of bats.
We're at Yellowstone Lake State Park in Wisconsin, and we're here to do some citizen science bat monitoring. And like many of the state parks in Wisconsin, this park provides a lot of great habitat for bat species that we're particularly interested in, such as the little brown bat, which is one of the bats greatly affected by white nose syndrome. And there's several bat houses here with 4,000 bats. When the bats come out at night to feast upon the very abundant insects that are here today, we'll be there with our bat detectors to record them and see what species we have. And we do an emergence count, watch the bats as they leave their roosts and count them. Every year I get more and more people coming to these events with interest in bats. They hear about white nose syndrome and they want to help to preserve and protect the bats that we have. Acoustic monitoring is where we take a device that allows us to take an ultrasonic bat call and brings it to a level that we can hear, like little clips and chirps of bats, and we use that information to get a relative idea of bat populations. And the data that we are collecting is important because in the unfortunate case we were to get white nose syndrome in Wisconsin, we'll be able to look back at our historic records and see where species were occurring, and if there were efforts to bring a species back, we'd be able to see where those species originally occurred. With all the bad that white nose syndrome has brought to the bats, one good thing that's come out of white nose syndrome is the increasing awareness of the public about bats and how important they are. For example, the Wisconsin Department of Natural Resources hosted a Wisconsin Bat Festival. Multiple agencies were involved in helping the public see what we do as far as research and what bats we have in the state. We even had bats from other countries there in a live bat display to get people up close with these bats to see that they're not the scary creatures that we all think that they are. And the cooperation from the public is the perfect partnership to protect the bats and prepare ourselves in the unfortunate case if white nose syndrome does come into our state.
Wisconsin, which is the last stronghold of little brown bats, 28 feet below us right now. 143,000 bats live here at Rita Mine. We haven't found white nose syndrome in Wisconsin yet, but we certainly have white nose prevention plans in place for every site that we have in the state. We sent postcards to all of the landowners within a 2-mile radius of our 2 largest hibernation sites, mainly in the hopes of just allowing people the opportunity that if they do see bats on the landscape during the middle of winter, to contact us directly, because the earlier we know, the better management actions we might be able to take. To be honest, I have to remain optimistic that every year we're going to go in there and not find it, but right now seeing as close as it is, there's certainly a good chance that we might find it within the next 5 years.
You know, anyone who
how adaptable they've been for the eons that they've been on the earth. And they are a tough animal, and if we could help them figure out what it is, they might keep them up. They'll respond. It takes time, but they'll respond. They are adaptable. We're not in the business of throwing in the towel. And we know it's too important. We know that we're the ones that are needed to do this, and we're going to keep plugging away at it. But we're not going to.
You definitely have to love the optimistic aspect for that last speaker on the video. So let me figure out how to stop this. I'll stop it down here first. So this is an excellent video, and please feel free to share it with whoever you wish to share it. Okay, so next up, I want to thank you very much for coming in and seeing the presentation on white nose, and I really appreciate the fact that people are interested in this. And if you, as a biologist with Cal Fish and Wildlife, is interested in assisting with this North American bat project, would you mind waving your hand, Scott? Scott is the one that's going to be doing the ground coordination on all of this work, so you may be seeing or hearing him at some point. He's a nice guy, so please be nice to him. And he's trying to do some very good stuff. So what would you like to do at this point? Okay, excellent.
So we'll go ahead and ask for questions in the audience first. Anyone have any questions for David?
Covered it that well.
All right. Oh. Hold on one second so the people on the internet can hear you.
I knew you had to ask a question, Daniel.
I just want to know how well is the artificial hibernaculum working that the TNC put out?
I believe it's only been in the ground for like 2 years, and it worked. I think it was last year they were reporting it worked well. They cleaned up the area. I'm not even sure. I don't think they detected PD there. I don't think they detected it. But it provides a potential safe roost for hibernating bats, and there were a number of bats. And you know, I hate to be anthropomorphic about this, but you know, bats sort of tell each other, "Hey, it's a pretty good spot." It seems like that because bat colonies, if it's a good one, they grow, and other bats come in from these other locations. So hopefully, it'll just continue to get bigger, and we'll be able to see what it does over the years. It's a little too early in the game.
Has anybody tried to disinfect caves? Is that something that's been attempted?
It's pretty much something that you can't do. Just there's so many nooks and crannies. We're talking about spores.
Yeah.
And yeah, it's pretty much almost impossible. Not to mention you're also going to kill all the cave fauna, which are probably just as equally important as bats are. So yeah, I have to say that bat biologists, if you were to talk bat biologists and say if we go back in time and go to Howe Cave and Severity Cave and Hales Cave in 2006, they would take the nuclear option and they would just destroy the entire ecosystem in those just to try to prevent this from happening. But we can't. We can't go back in time. So we have to just sort of face it the way it is. Another question? Yes.
Another question? Okay, hold on.
I was wondering if there was any updated information on the probiotics being used, and is that something they can spray inside the caves?
This is so new. I mean, we're talking about April 8th, this paper came out. So I think this is the new cutting edge. We were hoping we would find stuff like this. So over the last several years, funding has been provided through U.S. Fish and Wildlife Service grants, as well as Bat Conservation International and multiple other locations, Forest Service and so on, that provide grants for this research. And the research is still pretty much cutting edge. I think it's thrilling. I mean, 6 days ago is when this hit the thing, and I hope it spurs a whole bunch of other people to go, wow, I'm going to look at other things. Like, I'd really love to see California take this and run with it and see if it works for the diversity of of organisms that we have in California. I mean, we're the biodiversity hotspot. So have something developed here that could work against Pseudogymnoascus? I don't know, but that's what we'll hopefully find out. Other question?
Any other questions? How about any questions in your notes tab?
How do I tell if there's a question?
I think he said, yeah.
Click on notes. Oh, I see. Okay.
Okay.
Oh, wait, okay. Any questions from the notes tab? Nope, nothing. All right.
All right, well, thank you very much for coming. Yeah.
It was a lot of fun. Thank you very much.
I'm sure we did that.